Bismillah Rahman Rahim. Welcome, dear student. This is Biochemistry 2, course Chem 342 code. Today we are having lecture 20, and today's topic is isozymes, which is also known as isoenzymes. This is Dr. Naeem Khan, Associate Professor, Department of Chemistry, CUST. In today's lecture, we will talk about the isoenzymes description. We will highlight the important aspects related to isozymes or isoenzymes. Then we will discuss the different types of well-known isozymes. What are the differences between the different forms of the isoenzymes and how they are different and how they are isozymes. These will be highlighted. Then at the end, we will summarize the important applications of these isozymes or isoenzymes. So let's have a start. Isoenzymes or isozymes. One thing is very important, that the International Union of Biochemistry recommend that isoenzyme is more suitable word than isozymes. So we must note that isoenzymes is a preferable word for isozymes. So this term isozymes is better to be pronounced as isoenzymes. Isoenzymes are the enzymes. They are enzymes which are occurring in more than one molecular forms. It means that these are the enzymes which are the more than one different molecular forms. They are enzymes having more than one molecular forms, and these enzymes are occur in the same species and possibly in the same tissue and also possibly in the same cell. Isoenzymes are the molecular forms of the enzymes, different molecular forms of the enzyme which are occurring in the same individual, in the same tissue, or even in the same cells. They are enzymes occurring in more than one molecular forms, and different isozymes or isoenzymes catalyze the same reactions. Isoenzymes are the enzymes which are catalyzing the same reaction. They are occurring in more than one molecular forms, and they may be present in the same species, in the same tissue, or even in the same cell of the individual. Isoenzymes possess different kinetics. Isoenzymes are different from one another based on their kinetic characteristics. For example, Km, Michaelian constant, Michaelis-Menten constant, Km values are different for different forms isoenzymes. Also, isoenzymes are different from the regulatory properties. Isoenzymes have different types of regulation characteristic. Regulation means for the enzyme, it is very important that their activities must be tightly regulated. I mean, these enzymes are such molecules that they should be active whenever their activity is required. Whenever the enzyme activity is not required, it should be stopped, because the human body under different conditions require different things. Even after nutrition and the need of the different enzyme become active, because in the food we take different things. So enzyme must be active so as to digest the food. But these enzymes, digestion enzymes, are not supposed to be active every time. So, like the other enzyme, isoenzymes have regulatory properties, but it is important to note that isozymes have different regulatory aspects. They can be regulated by different routes. They may be similar but not identical amino acid sequence. Enzymes are made up of amino acids, and the isoenzymes are different molecular forms of the enzyme. They may be having similar but not identical. At least they will be different in the sequence of amino acids. Isoenzymes can be separated by electrophoresis. Actually, the amino acids sequence is different, and amino acids are charged particles, and some are positively charged, some are negatively, some are neutral. So on this basis, they have different charges. Net charge separation is the sequence of amino acids are different. So they can be separated by electrophoresis technique. Once again, isoenzymes are the different molecular forms of enzymes. They catalyze the same reaction. They are different from one another based on their kinetic properties like Michaelis-Menten constant, and they have different regulatory properties. They will be maybe having closely similar but not identical amino acid sequence. From a historic point of view, isozymes were first described by the Earl Hunter and C. Margaret in 1957, and the definition they give was different variants of the same enzyme having identical functions and present in the same individual. Different variants, different varieties of the same enzyme having identical functions and present in the same individual. This is a very broad term definition, because this definition includes enzymes. They are produced of they are produced from the different genes, and thus represent different loci known as isozyme. And enzyme, they are produced by the different alleles of the same enzyme, same gene, called allozymes. Though allozymes and isozymes are different things, but under this definition, they are considered the same. And in this definition, actually at 1957, there's the early stages. The isozymes was defined by for the first time by Earl Hunter and C. Margaret, that these are the different variants of the same enzyme having identical function and present in the same individual. Isozymes are closely related forms of the same enzyme. Yes, they are closely related, and the amino acid, as we saw, may be similar but not identical. They are share common evolutionary origin. They are originated through from common ancestor, and gene duplication usually give rise to the isozyme. That part of DNA where gene is located gets duplicated, and then it gives rise to different forms of enzyme. This gene duplication is responsible for the production of isoforms of enzyme. Allozyme and isozyme are used interchangeably. This is somewhat like a confusion. However, isozymes are the enzyme produced by the different genes, and allozymes are produced by different alleles of the same gene. The same allozymes are coded by different alleles of the same gene, while isozymes are coded by different genes. Both allozyme and isozyme catalyze the same reaction. This aspect is similar. Different isozymes of a given enzyme reflect at least 4 factors. When we are talking about the different isozymes of an enzyme, we must consider at least 4 factors. One, different metabolic patterns in different organs. Why the different isozymes present? Because the different organs have different metabolic patterns, so that's why different forms are required. Different location and metabolic role of the same cell. Even um, in a cell, different points have different metabolic roles. So that's why different forms are required. Remember, different forms can be regulated differently and they have different kinetic properties. Different stages of development. Different stages of development have different needs, different roles, different functions. And these different forms are therefore existing there. And different responses of isozyme to allosteric modulators. Yes, allosteric modulators are the molecules which are actually regulating the enzymes. So actually different areas, different locations have different allosteric modulators, and that's why uh, different isozymes are there, and that's why uh, their responses accordingly different. Giving some examples of the isozymes, one and the most thoroughly studied isozyme is lactate dehydrogenase. And LDH, lactate dehydrogenase enzyme, is the one which is catalyzing the reduction of pyruvate to lactate. It's a very important reaction. Actually, when there is the shortage of oxygen, instead of pyruvate passing to the Krebs cycle, it is anaerobically reduced to lactate, and in the same way, the reduced NADH coenzyme is oxidized. So this reaction in which pyruvate is converted to lactate is catalyzed by lactate dehydrogenase. This is an enzyme, and we are discussing for the isozymes. This enzyme has multiple forms called isozymes. Actually, this enzyme is made up of two types of polypeptide chains, and they are represented by M and H. Actually, the enzyme is a tetramer made up of four polypeptide chains of amino acids. And the five possibilities for the two chains are M4 when all are four are M type, M3H, M2H2, MH3, and H4. This M and H is basically on the basis of the existence, their predominant existence in the body. M are those which are predominantly present in the muscles or in the liver, and H are those which are predominantly present in the heart muscles. And this combination, we are having this five combination: M4, M3H, H3M, H2M2, H2, H3M, and H4. In this way, all M4 and M3H are predominantly are supposed to be occurring in the muscle and liver, and the H4 and H3M are predominantly supposed to be present in the heart muscle. So this is a isozyme because this enzyme is occurring in the five forms, basically made up of two polypeptide chains which are coiled upon itself in such a way that a tetramer molecule is made up of five forms are there. The characteristic of different forms of isozymes are like this. They are designated at LADH1, 2, 3, 4, 5, and this is combination is I saw in the earlier slide. H4, H3M, H2M2, HM3, and M4. And going to the location, H4 is mostly present in the heart and erythrocyte. This is again in the heart and the erythrocyte. And H2M2 mostly present in the brain and the kidney. While M3H and M4, skeletal muscle and liver, skeletal muscle and liver. Electrophoretic mobility, how they perform on electrophoretic plate. This moving fastest, this is faster, this is fast, this is slow, and this is slower. It means the speed is moving in this direction. And how they are affected by heat, 60 degrees centigrade have no effect on LDH1, no effect, that is partially affected, and LDH4, LDH5 are affected by heat at 60 degrees centigrade. And what is their serum concentration? 25%, 35% LDH2, 27% LDH3, 8% LDH4, and 5% LDH5. From this data, we can conclude that actually the five isoforms of the LDH, lactate dehydrogenase enzyme, they have variation in the concentration and the serum. They are existing in the different parts of the body, and they have different composition of the polypeptide chains. They behave differently on the electrophoretic plate, and they also have different behavior towards heat. These are isoforms and or isoenzymes of lactate dehydrogenase, which is catalyzing the important reaction of reduction of pyruvate to lactate. Another type of isoenzyme is the hexokinase. Hexokinase enzymes are also isoforms. Hexokinase catalyzes the first reaction of glycolysis. This is another important reaction because it is the regulatory reaction in the glycolysis pathway. We know that glycolysis is the first pathway of the carbohydrate metabolism. All carbohydrate must pass through glycolysis. So hexokinases are the enzymes which are catalyzing this first reaction of glycolysis, and that reaction is when glucose is phosphorylated to glucose 6-phosphate. Carbon number 6 is now linked to phosphate group. Glucose, we get glucose 6-phosphate, and in this reaction, 1 ATP is consumed. This reaction is catalyzed by the hexokinase enzyme. No, hexokinases are four types. Four isoforms are present in the mammals, and these are designated by hexokinase 1, hexokinase 2, hexokinase 3, and hexokinase 4. Hexokinase 4 is very commonly known as glucokinase, and the rest 1, 2, 3 are just represented as hexokinases. Hexokinases 1, 2, and 3 have low Km values compared to the glucokinase. Michaelis-Menten constant value is lower for the hexokinase 1, 2, and 3, while that's higher for the glucokinase. Glucokinase is not inhibited by the glucose 6-phosphate. The product of the reaction which is catalyzed by hexokinase is the glucose 6-phosphate. Glucose is phosphorylated to get glucose 6-phosphate, and this is having inhibition for the hexokinase 1, 2, and 3, but glucokinase is not inhibited by this. So this is a difference in the kinetics and the regulatory points of view of the isozymes. Glucokinase has lower affinity for glucose, but the hexokinase 1, 2, and 3 have high affinity for glucose. These are the differences and showing their different isoforms of the same enzyme catalyzing the same reaction of the phosphorylation of glucose to glucose 6-phosphate, but the isoforms have different characteristics. What are the important applications of this concepts of isozymes? Why it is important? First, isozymes in different organs allow the tuning of metabolism. In different organs, the different isozymes shows they have different patterns of metabolism, and this helps us to evaluate and to tune up the proper metabolic polarism there. By looking to the different catalytic nature and different regulatory nature of the different isoform, we can conclude and we can evaluate the metabolism in that organs. So this is helping us to tightly monitor the metabolic patterns and different tissues where different isozymes exist. Isozymes give particular needs of the tissue and development stage. By looking to the different isoenzymes in the different tissue or development stage, we can conclude about the different needs of the tissue or different needs of the development stage. In the development stage, if one isoform is there and then a next one appears, which means that in a proper stage, in the specific stage, they have different needs and different substrates and different environment, and that's why different isoforms is there. In the same way, in one tissue, one isoform is there, in the other another isoform is there. This also gives an information that actually there is some different needs. That's why different isozymes present. Measurement of isozymes, the test of various illnesses. This is a very important applications of isozyme. The measurement of isozyme levels is giving an indication of the medical illnesses. How? Especially if the lactate dehydrogenase H forms are more, this indicates that there is something wrong with the heart. Or if there is if there are high levels of the M in the blood serum, it would mean something wrong with the muscle. So this is a diagnostic test. Isozyme enhance the survival of backup survival as backup copy of essential enzyme. Actually, in the forms of isozyme, there are so many copies of the enzymes available. So actually, they are increasing the backup. If there is something happens to one form, other forms are readily available, and especially this increases the chances of survival of an individual. So isozymes are important from the perspective because these can be used for the fine tuning of metabolism in different organs. This can be used to summarize the needs of the different tissue developmental stage. This can be used as a diagnostic system of medical illnesses, and these are actually enhance the survival chances as they are providing backup copies of the enzymes. Dear students, Western Todd, GLJN, and Leninger books are recommended. Please highlight and thoroughly study each of the points to be discussed today. And from today, I will say goodbye. See you next time. Until then, Allah Hafiz.